So Friday's is discussion of things we have already studied. Let me take this out. This week, and there are questions. Okay? So I got 3 questions, and it seems that there's some question by email from somebody that is sick. So the first one is a little bit more technical about, not very technical, anyhow, but it's asking to explain for the case of Lotka-Volterra. Remember here you had the prey, and here predator, or was it, no, contrary. Prey, predator, and we had this null clients which in the units that we are using were 1, okay? So the fixed point is 1, 1. So the question is, why does it imply circles or oscillations, okay? So we had this, we have drawn this kind of arrays here. Okay, so okay, this you remember, yes? That the null client is where the populations are 0 for this the null client for preys and predators. When you cross the lines, you go from a positive value of the derivative to a negative value of the derivative, meaning on one side you are increasing, the other side of the null line you are decreasing. Okay, so okay, let's start here. And why will it be related to oscillations? Okay, so let me draw this in color. So this goes like this, down and to the side. Okay, so it should go something like that. Okay, but now when you cross this point here, you have to go up, but continue to go this way. Okay, so you go up, but you continue this way. Okay, by going up, you will eventually cross this line. Okay, so now you could continue to go up, but you should go this way. Okay, and when you cross again the line, now you have to go down and this way, down and this way. Okay, so obviously my drawing is very bad, but yeah, but you see that you are first the prey is decreasing and the predator is increasing, then the prey increases, decreases, increases, and decreases, and so this creates this form of circle here, okay, which as I told you, I did not show, we cannot show here with only this reasoning that it's really a circle or something that could go like this, okay? Or it's not even a circle, it's a closed curve, okay? So what we could not show that it's closed just by this reasoning, okay? Anyhow, but we could think about this at least near the fixed point when we calculated the famous eigenvalue, which was an imaginary number. So if the real part is 0, it's kind of the oscillation with constant amplitude. There's nothing that makes the increase and decrease the amplitude of the oscillation. So you would expect, at least from this other, more quantitative reason, that you could say, this should be really a circle, at least near the fixed point. Okay? Now, the whole diagram that I have drawn with all the curves like this, with this obviously cannot be inferred from a local stability analysis. This is called the local stability. It's the stability of the 1, point. Okay? Now getting the whole thing for initial conditions, they are far from this. It's a global analysis which we have performed. And I mean, not very well, but we found that famous conservation law, and then therefore they could infer the closed curves. Okay? Yeah. But this is difficult to do in general. Okay? So I think this answers the question. Yeah. Mike, where are the mic? Oh, okay. Okay.
No, it's just that I believe that it's in the wrong direction, because in that
again,
way,
the wrong direction.
yeah, because in that way the predator will increase and then the
okay.
prey
I
increase.
again changed the
you need
okay.
to change
Okay.
the axis just.
Okay, change the axis again. I'm not good with axis. Yeah. Okay, so okay, there's a new yeah, I have to which relations plus minus we can't describe by predator-prey modeling, like parasitism or amensalism, and why? Well, if it's plus minus, it's predator-prey-like, okay? So the point is, for instance, parasitism is also described usually by kind of predator-prey models, but usually, if this has to do usually with insects or yeah, usually insects or small animals, then this is usually modeled with difference equation, with not differential equation, but you know the mapping, the discrete time that we developed in the third class last week. So that this non-overlapping generation case, but you have the discrete time, and the time is counted in generations. Well, then parasitism is usually modeled in this context, and then it's also a kind of predator-prey model. It's called the, can't remember now the name. It's a system of two different difference equations. I can't remember the form of the equations. You have a prey, you have predator, or parasite, and so on and so on. Parasitoid, for instance. Okay, so that is usually the way you model this. Plus minus is always some kind of predator-prey dynamics, okay? Because we, as I told you, we are using a predator-prey, the word predator-prey dynamics for antagonistic relations in general. Is the metabolic rate implicit in this manipulation variable we included in the Rosenzweig-McArthur model, or could we somehow include it? By metabolization rate, which is actually metabolic rate, I mean the time it takes for the predator to incorporate the food and be hungry again. What would be the effect of that in those simple predator-prey models? Yeah, so metabolic rate is something related to the individuals of the species. We are modeling the population. Okay? So sure that parameters that appear in the equation, when you should reflect, ideally should reflect things that happens with the individuals, and then you scale from individual to population size. Okay? That's in the case of predator-prey models that we used. It's called mechanistic modeling, okay? And it has something to do with the metabolic rate, which would be, if you would like to use the metabolic rate, and somehow this should be a modeling of the of how the manipulation time is influenced by metabolic rates. Sure that that has to be some relation, okay? Okay, but this is not a modeling, a populational modeling anymore, okay? This is how the parameters are determined by the species metabolism or physiology and so on. And at the end, at the population level, you will probably get that the certain parameter should be in a certain range of values. Okay? So, sure, but at the population level, we don't have metabolic rates exactly. Yeah, the phone, yeah, the mic.
Okay, so I wrote a question because we included this manipulation variable, but that's at the individual level, isn't it? I wasn't really quite sure about the nature of this manipulation variable.
Yeah, okay, so we can, okay, how is the way that usually we derive this expression for the what's called the functional response, okay? So just, okay, I have it here with me, actually, so let me see if I can remember this. Okay, let me see. So we usually have something like this. Let me, d and dt, okay, would be Rn 1 minus n over k. That was the logistic growth of the prey. Then you had something here, which is proportional to p times n. And in the Rosenzweig-Makar, we have this here. You have a c. You can have c times some value tau times n. Okay, so that's the functional response. Okay, if I have something like this, just p, some function of n, or n in p eventually, this is called the functional response. And this is called, and there's a classification of the most common kinds of functional responses. It's called the classification of Holling. And this is Holling type 2. Holling type 1 is just linear. And then there's a Holling type 3 also. Okay? So, well, and the equation for the predator is some other variable here that b p n 1 over c tau n minus some death rate of p. Okay, so that was the Rosenzweig-Makar model, and we go back to this term here and see how this is related to the actual value, the fact that you have a manipulation time. Let's see that, okay? It's almost mechanistically derivations. It's called in ecology books, it's called mechanistic derivation, which I will show, although it's not, it's a little bit hand-wavy, actually. It's not very formal, okay? So, okay. Therefore, let us remember that term here, okay? Is if I have one guy, okay, one predator. This term here, when you look to the effect here, the derivative is the number of prey consumed by unit time by one predator. Okay, so we want to calculate the number of prey consumed by unit time of one predator. So the derivation goes as follows. So consider. Okay, let me see. Consider that in the time that a predator spends searching prey plus the time it manipulates, okay? And this is a kind of manipulation sensu largo, which means digestion is also manipulation in this case. You say, okay, yes, he already ate his prey, so it's not anymore neither searching nor manipulating, but that would be really manipulating in the precise sense of manipulation. So this manipulation is, okay, he's still digesting, he's satiated, and so this counts as manipulation time. Okay? So now, the time he spends manipulating should be proportional to, say, the number of prey times some unit manipulation time, okay? So if it spends 1 hour, okay, manipulating each prey, if he has consumed 10 prey, he spent 10 hours, okay? I'm assuming linear, right? So, on the other hand, the number of prey consumed should be some alpha proportional to the number of prey consumed. Okay, let me number of prey consumed. Okay, let me put an n, small n, because this is not the total population of the prey. So the capital N was usually the total population. So the number of prey consumed should also be somehow proportional to the number of prey which are available. So the total population size of the prey. So if the population size is large, it's a big number, then it's easier to find the prey. So should be somehow number of prey consumed per unit time. Okay. Per unit time should be the total, proportional to the total population, but also somehow proportional to the time he spent searching. If he searched longer, then he will also increase the number of prey that he could consume. So now, the total pop time can be written like Ts, and Ts is n over alpha n, okay? Plus T hash, which is just n hash, okay? Which is equal to n times 1 plus hash alpha n over alpha n, okay? Agree? Okay. I put the n in evidence, then I get this gives you the hash here, and this is this one.
Professor, shouldn't be like the opposite if TS is bigger, like more time searching for a prey, the consumed number of prey should be smaller?
No, the idea is that it should be proportional to those things. If I spend more time searching, I will find more. Okay.
Ah, okay.
Okay.
I thought
okay.
Was
For a given population size of prey. Okay. So there's a certain number of prey available. If I search longer, I can find more.
Oh, okay.
Okay.
I was thinking it was like search time for each prey. It's like total searching time. That's
the time it takes to search. He spends searching. Okay.
Yes.
And this one is the time he spent manipulating. Okay. There's an elementary time here. Okay. But this is the life of this predator is divided between searching and manipulating. Okay, so very simplified thing, but anyhow, that's what is in this model. So now it's easy to see that the number of prey divided by time. Here I made a mistake. That was the number of prey consumed. Okay. Number of prey per unit time. Unit time is just 1 alpha n over 1 plus alpha h n. Okay. So sorry, I made some confusions during the improvised demonstration. So this is the number of prey that's consumed. It's proportional to the time he spends searching. Is proportional to the number of prey he consumes, and times an elementary time associated with the manipulation. And here, the rest is just a mathematical manipulation, which says that the number of prey consumed per unit time is something like that. So, and here's the time, Ash. So it's connected to this time of manipulation. Okay?
Professor, here.
Yeah.
Alpha is the efficiency
In this case, alpha is
catching the prey.
it's not the efficiency. It would be the attack rate and the absence of the searching of the manipulation. Okay.
Okay.
Okay. So with the Lotka-Monteiro model, we had to have some GNDT equal, that is something, and then there was minus alpha NP. Okay.
Okay.
Yes. So this alpha is the attack rate. Okay. It's the number. But this in this case, it's also the attack rate with the attack rate without the effect of the manipulation time. Okay. So that appears here. Okay.
Okay. So getting back to the
Now.
okay, now it is. So I say that tau in the original equation is just the interval between meals.
Could be, yeah. That is, I mean, it's this general manipulation time, but it's in the sense that, it could be, if it's really a predator-prey in a typical sense, it is the time between meals, yes. So, okay, so this is connected to, that's why the Holling type 2 functional response is connected to manipulations. This is the argument, the general. It's a little bit hand-waving argument, because you are modeling the behavior of the predator as being simply spending time searching and manipulating. Okay. So this could you could and other activities. Okay. So they could have an typically if in certain situations there's also interference between the predators. Say if the number of prey is small. Say if the number of prey is concentrated in a certain place, we have problem with areas. So there could be a time spent with the interfering with the others, just scrambling for the others, giving some kicks on your co-specifics. So you could also have something in time here of interference, which is a kind of competition, okay? Yeah. You could have this, and then you could do something similar reasoning, and then you would have something like this, plus some new time, Ash, like, okay, G alpha P. Okay, you would get this. This is a new functional response. It's not the Holling type, because that depends on both the predators and the prey. This other one depends only on the prey, okay, the one I just wrote for. Now this depends on predators and prey. Okay. So that's the Beddington De Angelis. Functional response of Beddington De Angelis. Okay. So, okay, that's okay. I think that's enough for this question. Yes. Okay. Okay, this 3. Now, how the determination of parameters actually happen? Does it take a lot of time and money? Right. So we usually do some renormalization and change the variables in order to reduce the number of parameters. Does it help only to study the system mathematically, or the new parameters, generally less parameters, can be easier to determine, maybe easier to determine experimentally? No. The parameters that you can eventually determine experimentally are the parameters that have a clear sense in the original equations, and then you go and you take non-dimensional equations, and then you simplify the system. You have less parameters, but you have combination of parameters, okay, that are that still are in the equations that you want to study, say either computationally, or you want to study the general qualitative dynamics and so on. So this is a mathematical trick, and when you get to the end, you could say, okay, now I go back to my original variables. The physicists would call it the physical variables, okay? But it's not physics, the biological variables in this case, okay? And that's the one that you could imagine you could determine. Now, does it take money or time to determine parameters? Well, that depends on the species. What are you trying to look for? So, which parameters are easier to determine? Usually, well, something that we usually know is the intrinsic growth factor. Okay, so R in the equations. Okay? So this is the one that can be measured. So there are parameters that depend on interaction, and there are parameters that depend on the species. Okay, the ones that depend only on the species are usually easier to determine, but the ones that have interactions are much more difficult. Okay, so for instance here, okay, let's forget this guy. You could try to do some experiments to determine the attack rate and maybe also the manipulation time. Okay, more or less. But for instance, the efficiency of transforming prey to predators, how many prey correspond to one predator? I mean, how many preys are consumed will give rise to one new predator. That's the efficiency. That's very difficult. Okay, so this kind of thing can be done in the lab with the microorganisms like protozoa or plankton or something like that. But in the field, no. Usually you don't know. So the problem of parameter determination is a big problem. And when you say that you get, let's say that in a more general way, not only for predator, prey, and so on, you have a system that you are modeling, a biological system. It has lots of equations, lots of parameters. Parameter hell, again, okay, if you get it. Then what you usually try to do is, let's see which ones I know, okay? So an example which is out of the context of this week class, but I've been working with modeling COVID-19 epidemic in Brazil over the last 2 years. And then we have models, which are complicated models and so on. And then you have to calibrate the model. So what are the things that you know and what are things you don't know? There are some things that like the time a person typically spends infectious. So this is usually something you know, more or less, the time it takes that you are in the infectious period, okay? This is something that people know, more or less, okay? So for COVID, it would be more or less a week, okay? So you already have an idea, okay? Like a week. It's not a year, okay? So you don't know, you already know something, okay? You know the scale. So then you use this thing. But then there are parameters that you cannot know, which is the, say, the probability of, if you have an infectious person, a susceptible person, that an encounter of them will generate a new infected person some days later, okay? This is not observable for respiratory diseases, okay? That's not impossible. Even for respiratory diseases, it's difficult to say what is an encounter, okay? I mean, you can get, sometimes you go a closed room, you go a bar, lots of people speaking, and even if you stay away from the people, you have been in contact with the others because it's the place. On the other hand, maybe you are in a group of 10 persons, but you are in open air. Maybe this does not count in this case as a count. So it's very difficult to know what's a content. Okay? Then, even more difficult would be the transmission rate. Okay, the probability that so this is usually a parameter that, when you have the data, this is the parameter that you will fit. Okay, this is the parameter that you will fit. And you try to use as many other parameters that you know. Now, this brings me to a topic that is also very important. For instance, you could say, okay, I have my, let me talk in time here, number of new infections per day or per week. So, and then you know that this is something like this, and then the variant comes, and then it oscillates and it goes, and so on. All the stuff that you read in the newspapers those days, or in the television. Okay. Then you say, okay, I have a model, and I want to fit the model to this curve. Okay. My model has 20 parameters. I will do a model fitting with the 20 parameters. What will you get? You will get exactly this curve. Yeah. Why? But if you have so many parameters, you can always fit everything. Okay? But you are taking this curve too seriously. Okay? Because it's approximate also, the collection of data and so on. So that also represents the fact that this system has noise. And what you are getting is the notification, the cases that have been notified, that have been somehow entered into the system. It took time to enter the system. If you look this day by day, it's very curious, but there is always a small number of new infections on Sundays. But this is a problem of the health system, that the people that do the notification don't work on Sundays. Okay, so better take a week as a unit time for that, because a week repeats itself clearly. Well, there are holidays and so on, but you know. Okay. So the point is, you cannot overfit models. You should never overfit models. Okay? So what the best practice would be, I have more parameters that I have some idea, a range of values. Okay? Then I choose I run the model computationally, choosing the values for these parameters that I more or less know, and then I run it many times, say certain parameters between 0 and 1, then I take, say, first run 0.5, but then I try with other values also. So this is a sensibility analysis, and if your results change a lot, then your model is problematic. Okay? It depends on very fine tuning of things. Okay? But if your model is okay, then maybe things don't change so much. Okay? And so that's important, and not only obviously with COVID and so on. It's even if you do a polynomial fit of any data. Okay? So let me see here. I have some data, some value, some have time, and I have some data here. Okay, of anything. Okay, and I got some points. I've got this. Yeah. Now, how many I have? 1, 2, 3, 4, 5, 6. If I use a fifth order polynomial, I have six coefficients of a polynomial, and I can fit exactly. I can fit exactly. Okay? Now, this is taken that these values here are really like very deterministic thing. Okay? No noise in the system. And then you say, okay, now I want to make predictions, and then, okay, this polynomial does this. Okay? And then you, after this point, then you go and you say, okay, let's see what happened, and you got something here, here, everything was wrong here. Okay? Why? Because this is a system with noise. Okay? So better try to fit what? Here, a line. Okay? Because it's more or less increasing, monotonically increasing. Okay? A line has only 2 parameters, and then you can get a good fit. Okay? And that's okay. Okay? So you use like least squares or whatever you want. Okay? So you don't want to fit exactly. When you are overfitting, when you do the parameter, 5th order parameter, they are overfitting the system, and actually you are not finding what's important for you in the data. Okay? So that's the same thing with, instead of polynomials, also with differential equations, which have a lot of parameters floating around. Okay? So, okay. So there was a question by email, yes? Okay, let me see. Okay, it's not the best moment to have a slow internet connection, yes? I didn't bring my computer today. Okay, I didn't get the yeah, maybe you could read the question.
Hello. So from Thales, is there any experimental verification, like using microscopic organism petri dishes, of the enrichment paradox?
So you can have experimental, not confirmation. Right? First, enrichment paradox is not a paradox. Okay, people. No, the paradox is conceptual in the sense that I enrich a certain. What's the idea? I will enrich a certain environment. You give food for one of the species, in this case, the prey. Okay, so I give a lot of food, and instead of having a super stable, being good for the prey, it can actually crash the population and eliminate the prey, and the predator, if it's a specialist, goes also away. Mutual elimination. Okay, so this would be the paradox in the sense I give more food, but they die out because they give too much food. Okay, so with microorganisms you can do a lot of things, and you sure they can do this kind of situations. I don't have in my mind anymore. You have seen this years ago, some paper, but you couldn't do this. Okay, but the only point is okay, because I can manipulate with micro parasites and microorganisms, and I can have really 2 species case, for instance, resource in 2 species. I can almost engineer things so that the species will solve the equation for me. Okay, more or less. Yeah, it's like that they will obey it because I'm manipulating in such a way that it will happen. Now the point is how relevant is this for ecology in the field? And this is obviously a big question mark, because it's there are crashes, we see crashes, right? But well, sometimes is it enrichment paradox or not? Well, difficult to say. Right? So there's a second question.
What will be the effect of including more species in the predator-prey dynamics, assuming like a predator that feeds on many preys and eats what is more available? I think about this related to what differences in predator-prey dynamics would we expect to see between the less complex temperate trophic networks and the more complex tropical ones?
Okay, so that's the topic for next week. Actually, so if I put more, okay, I go beyond that. So what we will study next week is consumer resource dynamics. So we could start with one consumer and one resource, but this is, I mean, that's too much similar to predator-prey. So we will start with having, say, 2 consumers and 1 resource, okay? And so we will draw diagrams like that. Consumer 1, consumer 2, resource here, the resource it's been consumed by both of them, and then we can write the consumer resource equations, okay? So this is a competition. That's resource competition, okay? And then you can, instead of using Lotka-Volterra competition model, which is phenomenological, this one is more mechanistic in the sense I'm actually describing the consumer relations, okay? Well, it will turn out that one of them will eliminate the other in this case. Okay, so we will see this, and this is the concept of competitive elimination, okay, that we have already discussed. Right? So, but then we can go on with for instance, we can modify this case and saying I have 2 consumers, and say usually one of them will be the winner. Okay, on the long time, only one of the consumers will prevail, okay? Let's say it's the consumer number 1, okay? So you normally would eliminate the consumer 2, but maybe consumer 2 is smart, and consumer 2 actually can prey upon the consumer 1. So there would be an array going this way, okay? So this is called intra-guild predation, okay? Guild is because these 2 here are a guild which is consuming this resource, and intra-guild, it's a predation between them, okay? So this you can have also in the lab. Actually, I even modeled with a student of mine intra-guild predation with mites, okay? Mites is, how is that called in Portuguese? Okay, so everybody knows what's a mite. A mite is ácaros. In Portuguese is ácaros. Okay, so there are several species of mites, and there are some that have intra-guild predation. Okay.
Professor, como funciona? How would work the dynamics of species that feed on dead animals, because they depend on the presence of the species they eat, the death of the species they eat? How it would work? Like,
yeah, I mean, then we have two possibilities. I mean, usually, for instance, here, okay, let's say that animal is the resource. Okay, could be other things, but okay. So there's something that has to be the resource replenishment, or the new resources. If I have only a certain amount of resources, the dynamics is this will collapse. Okay, so you have to feed somehow. Okay, so you can model the thing like saying, okay, there's a certain number of dead individuals of species that is the species R that appears over time. Okay. So this is reasonable if it's more or less constant. Okay? But then you could also say, no, this guy here actually is being comes from interaction with another species, which is it's species P here, which maybe consumes this, but only partially. Consumes, let's say, something else here, the same species but alive. Okay, so R bar. So this is the species alive. Okay, so it consumes this. Okay, it's not connected to this, but actually it creates an influx of dead because it does not consume completely. For instance, okay, then you can have a lot of things happening here. Depends on the assumptions of generalist or not generalist or whatever. Right? So anything goes. Okay, so yeah. These are called community modulus. Okay? A community modulus or community moduli. Okay? So, so we will take a look at this in the next week. Okay? So we don't have any more questions, but you can ask if you want to.
Okay. Well, I have a question related with data. When we have data of predator-prey behavior, but since a noisy cycle, how can we handle that noise? I mean, is it possible to include a variable, a random variable in this kind of models? Or
Yeah, you can. So, okay, you can do several things. You have data, and you try to fit the equations. Okay. So, and you know the data is noisy. Okay. Then you can do this by several ways. Actually, this was explained in the opening lecture by Mercedes Pascual. There's a famous program that's it's called POMP, partially observed Markov process. So you will have to make the hypothesis that your system is not deterministic, right? I mean, okay, let me see. You could do this simple thing because it's just least square splittings and so on, and that's it. You could do better. Doing better is assume it's a Markov process. It's a stochastic process, okay? And then you do stochastic fitting. You will see this with Ricardo Martinez in the next course after mine, okay, the stochastic modeling. And then you assume that there's noise, environmental noise, usually, but you can also have noise coming from the measurements. Okay, so you have the environment, the naturally varying things that affect a given population, because it's not isolated from other populations, not isolated from the environment in general. So you have noise because there are things varying. Okay? But then you can also have some more what would be an observational noise. Okay, so maybe you have difficulties with your measurement apparatus. Okay, and the apparatus is anything, can be traps in the field or whatever. Okay, somehow you have the measuring process, which is imperfect, right? And then you can model this sometimes as saying that, okay, there's my data. When I measure them, actually they are not very precise, okay? And I modeled this by introducing a noise that represents that the things I don't know really from the data, okay? So you'd have to think that this very different thing as a lab in physics or in whatever, in chemistry also, okay? Or engineering, right? Where you do manipulate everything and you have also noise. Everything has noise, but sometimes such a small level that you don't care, okay? So, but in ecological problems, also in epidemiology, in many things, everything that goes with populations and communities, you will have the environmental noise and also the noise that comes from your observation difficulties, okay? So, and that's why ecologists use a whole set of statistical machinery, statistical formalism. So, and typically an ecologist knows much more statistics than any physicist, okay? And unless you have, for instance, you could be in cosmology where you have also an observational problem, yes? When you go look to the sky, you cannot manipulate the sky. And then you have also problems with statistics of the imprecise things, and that there are stars that you cannot observe, but they are there. So you have a similar situation for physicists. Cosmologists know more statistics than the others. But usually ecologists do need to use much more complicated statistics than in a simple physics experiment. Really needed. Okay? So there are people that are specialists on this, and this is far from trivial. Right? So, okay, so the noise is there. In any realistic setting, you want to fit data, for instance, also with the COVID-19 things, where we also have to introduce noise and so on, and see if this is still, I mean, if some, because you could have, for instance, could have models that are structurally unstable, like the Volterra model. Like the Volterra model under noise has no cycles anymore. It goes up and down, but it doesn't close. You don't see the cycles, and not with noise. Okay, so if you model, you have a nice model, but you introduce noise, and it gives you completely different things, means that your model is not really predictive of anything. Okay? In this sense, also, the relation between data and theory is very different in ecology and in physics. Okay? In ecology, you usually have models, and then if the model somehow fails in a certain situation, you try to improve your model. Okay? So it's different from physics, where you can have precise predictions, and if they are not, you can say my theory is wrong. Okay? More or less. It's not so simple. Okay? This is a little bit too much Popper, but even Popper would not agree on that. But for instance, it's clear. Let me talk a little bit about relation theory and experiment in physics, which is not so trivial sometimes. So you know that the solar system is very well approximated usually by the equations of Newton. Newton mechanics, Newtonian mechanics, celestial mechanics, traditional things developed since Newton and even a little bit before. So you have the model of the solar system, you have the equations, and people went on and calculated the orbits. Then with the development of optics, they could have better and better measurements of the position of planets. And they could look the planets, look their orbits in the sky, and see if this corresponds to Newton's laws. Yeah, sure. Very nice. And one day this failed. Okay? Didn't work, because the orbit, I think, of Uranus was atypical, was not the one predicted. Okay? So, does it mean that the laws of Newton, the mechanics, classical mechanics laws, are wrong? Or not? Mike.
Usually in mathematics, when we are modeling something, we most of the times can do like put everything realistically in a model and put all the parameters and everything that we want to. So sometimes you have to kind of eliminate in some sense some parameters or some of the conditions. So we have a simpler model that we want that most of the time we works, but there will be cases that there won't be enough. So it doesn't mean that the model, the Newton equations are not true. It's just that it's a case that the model is not enough to model the situation.
Okay, on this line, yes. When you look for the orbit of Uranus, you're making two assumptions: Newton's law and the model of the solar system. Okay, you can modify Newton's laws, or you could modify the model for solar system. So it was Lagrange. This is 18th century, I think, 18th or 19. I think it was Lagrange, and so say, okay, let's suppose that there's still one planet more that we haven't seen. Okay, let me calculate the orbit of this hypothetical planet, and see if this could explain the movement of Uranus. But then obviously there's a problem that you are imagining a planet that you don't see. But then maybe if you calculate the orbit, you can, with improvement of optics, essentially, try to search for the guy. Maybe it's a very tiny signal. Okay? Yeah, actually, found a Neptune. Okay? So Neptune has been discovered in this way. Okay? So very nice. So the model was wrong. Okay? And the theory was still persisted. But then, when you go to the end of the 19th century, you have again an orbit that's not okay. That's not okay. It's Mercury. So the orbit of Mercury is not the one predicted by the Newton's law. Okay. So what did people do? That should be a new planet. Okay? New planet. Vulcanus. Okay? Never seen Vulcanus. Okay? Or maybe it's too close to the sun, and we don't know well the physics of the sun. Maybe there are heterogeneities that we don't know. Maybe it's connected to this. No real convincing mechanism has been found. Okay? So then comes Einstein and says, no, the theory is wrong. You have to use general relativity. Okay? General relativity substitutes Newton's laws. It's a new theory about gravitational interactions. Okay? So you use the new theory, general relativity, you calculate the orbits, and you get exactly the orbit of Mercury. Correct. Okay? So you have to change the theory and not the model. The model was okay. There were no volcanoes, nothing like that. And the model was okay, but the theory was not. Okay? Then you say, okay, but you use this to explain Mercury, but the other ones obey Newton. No, it's obviously that Newton is a limiting case of the general relativity laws, and when you are far from a very massive star, like very close to the sun, and if you go far away, the effects which come from general relativity are not even perceptible. So you can use Newton. So this is the kind of relation we have between models and theory. Now, in ecology, or more fundamentally in biology, you don't have Newton's laws. Okay? There's no or general relativity or quantum equation of Schrödinger and so on, which in physics we think this is the fundamental thing. Okay? So the closest thing would be Malthus, but anyhow, as we saw, Malthus is never true. Okay? Really, there will be always some interaction, because it's only valid without interaction. If you interact, it's already not anymore Malthus. Okay? So the theory in ecology has a different perspective. So you have a lot of conceptual theory, which are hypotheses of what are the mechanisms, which are the patterns that are important, and then you try maybe to provide a model, and then this model can be also conceptual, but can also sometimes be mathematical, like the ones we have here. Okay? So this is different from physics. There's no fundamental law. Okay? But what you do with the models, it's very different in ecology and in physics, for instance. Okay? In ecology, the things that have been shown here are models that develop concepts, and like predator-prey cycles or competitive elimination. And these models give you kind of framework to think about your system, your ecological system, about ecology in general. Okay, so this gives you a framework in the sense that I discussed before. Competition creates elimination. So if you see species competing, but not being eliminated, we have to explain the reason that they are not, one is not eliminating the other one, and then you create questions about why it's not happening. So you have to use the theory before. You have the theory, which tells you something, then you go and you see that there's something that's not really connected, really predicted by theory. But in ecology, there is no unless you have microorganisms in the lab controlled, and even so. Now, in general, there's no isolated system, which is the basis of the construction of physics. You isolate, you put in vacuum, you put a cold temperature, whatever, to eliminate all effects that would spoil your experiment. Okay, but there's no such thing in ecology. So in ecology, you have really what you could call a complex system. There are lots of species, lots of individuals, lots of communities. They are all interacting. The interaction sometimes changes. It's non-linear. The process is such that the interaction creates effects that will affect the interaction itself. Okay? So this kind of feedback mechanism which is nonlinearity in mathematics. So this creates the thing that there's lots of things connected. And so when you model, you have to somehow cut your interactions somehow. You cannot say the only thing I can do is model 10,000 species at the same time, okay? Because parameter hell and so on, okay? Yeah? So then you say, okay, I would model 2. But obviously, there's no such thing that the 2 species that they interact only between them. Okay? That's obviously not true. But sometimes, as in the case, for instance, of single species, we had a long discussion on when can we use the idea of single species. And well, sometimes it's good approximation. So it's always approximation. And these things are also approximations. We are doing models with 2 species. Well, you can then use them as building blocks for n species, for lots of species. Then you can ask questions about the interaction networks, how they depend on how the stability of a certain community depends on the number of species and all of this. So you can do this, and it's done. Much of this research has been done computationally, mathematically, but you have always to think that this is not the same thing as in physics. I make a prediction and go there and, okay, here, Neptune, okay. There's no Neptune in college. Okay.
Thinking about this, like nothing is completely isolated when we are
oh,
sorry.
No, it's working. Okay. I didn't understand.
Thinking about this, that nothing is completely isolated when we're thinking about ecologies and those systems. I was wondering about like interaction between 2 species, but like 3 species and animals that feed on these species, but they have different sort of interaction. Like this animal could eat the seed or could disperse the seed. How do you model these 2 sort of interactions between 2 species?
Yeah,
like
okay, so 3 species will be something like that, but...
No, 2 species.
2 species? But with different effects, yeah.
Yeah, they have, like the animal interacts in both ways with the tree. Like it's only 2 species, but how do you weight this in the model?
You have I mean, you can do this. That would be a mutualistic antagonistic model, okay? So, for instance, one animal feeds on the seeds of a certain plant, but it's not all feed, and it also takes the seeds around, okay, which creates space for new plants, okay. So it's mutualistic in some sense, but it's also antagonistic. I mean, you can do this, write this in an equation. Just put a mutualistic term, and then you need to know if the mutualism is obligatory or not, okay. So can the seeds be dispersed in another way or not, okay? Maybe they could just by wind, I know. Okay? So then you have to know if the mutualism is really important, okay? But usually if it's obligatory, it's obviously important. So then you would, I don't know the result of such a thing, but you can write this equation, so write the equation like you did with, I don't know, Rosenzweig-Macquarel, and put a new term, which is an increase of the population of prey due to the presence of the predator, and so on. Okay? Then probably you have extreme cases where one or the other are more important. Yeah, so I mean, you can imagine how to model this.
My question is about the modeling, the development of the model. For example, in COVID, we have a very difficult dynamics because there are government measures, there are vaccines, new variants, etc. But when we are doing models, we usually just add these parameters, and these combinations are linear, like linear, like we just put n or susceptibles or infecteds, and we never put like square or two-third. And how can we know that this is the best model?
So, okay. So that would be like there is no functional response other than the linear in epidemiology, okay? Kind of. We usually have in epidemiology, for instance, number of susceptibles is equal to, can only, okay, let's say there's no birth, okay? Can only decrease, and then usually some beta susceptibles times infected, then the number of infected increases with the same coefficients because it's 1-to-1, one infectious gives, then there's some deaths, not that, and people go out of the infectious stage because they are okay and they are recovered, and you have the recovered that you usually would be just the guys that recovered from the infection. So this is a typical, this is SIR model we'll see in the last week. Okay, but so now why this? Okay, that's the question. Why this? Couldn't it be smart? Yeah. Okay, or here. Good. Well, okay. So most of the time, data in epidemiology are very, very, very noisy, and you maybe not even distinguish between the forms. Okay, so this is one possibility. But actually, together with some collaborators, I wrote a paper with some modification of this, but it's a long. It's about malaria. Okay, so let me remember a little bit. So if you look at this, the number of susceptibles being infected is proportional to S times I, but if the number of, okay, in malaria you don't have this. Okay, you have something like ds dt is equal to beta s times, but I will call I index m, infected mosquito. Okay, so we have mosquitoes and non-mosquitoes. Okay, so actually, when you have a very small number of mosquitoes, okay, then you don't expect them to be able to bite very efficiently. Okay, so usually this has some different form. I can't remember the form now. Okay, so they can have some effects of small populations where you have to modify these terms. It exists. For instance, this is used when malaria, in the case of Brazil, most places where you have malaria, kind of endemic situation of malaria, which is not the same as in Africa. Okay, in Africa you have usually the mosquito is called the Anopheles falciparum, and here it's vivax. So different species, and the ones that are most prevalent, there's also falciparum, but the largest part is this vivax mosquito, and the disease is not so severe. Okay, but can also be anyhow. But you find this the most. Where will you find areas with malaria? Okay, so you have to have people and mosquitoes. Okay, therefore, if you go really to the center of the Amazonian forest, you don't have malaria because you don't have people there enough. Okay, so there's no chain of connecting because there are not enough people, and so therefore You can have a case or two, but will be just no chain of so you can have cases, but you don't have epidemic. Okay, but okay, then you go to cities with good infrastructure, and there's no way the mosquito can live there. Okay, and so where will you find it? For instance, in Brazil, it's the border of the forests, where you have people, usually illegally occupying terrain, and then you have also still a lot of vegetation, and the mosquito is able to adapt to this perturbed vegetation, and then you have malaria cases in these regions, like in Acre, for instance, or you have this kind of thing, which are the frontiers of contact with where you have people and where you have the forest. So that's the most common cases in Brazil, and that's not in Africa, because you have much more cases which are severe in Africa, and a lot of people die actually of malaria. Okay? So how many people do you think that die in average per year in Brazil from malaria? No idea. It's like 10. Okay, yeah. Not the cases that die from it. Okay? So the severe, I mean, there are severe cases, but people recover. But who really dies, it's usually comorbidities and so on. So it's not it's a burden, because it's a disease that requires treatment, and that therefore there's a burden, a financial burden and a health burden and so on. This is important to eliminate. But it's not only because people die, okay? Which is one of the things that is very badly discussed about COVID. It's not only that people don't die of the new variants so much, okay? But still a burden. You have the people hospitalized, you have a lot of things. Even if they survive, it's not good to have a disease. Yes? So it's like 5. And what do you think more or less of dengue? How many die from dengue? I think in 18 or 19 was like 800. Okay, it depends a little bit, because there are years where you don't have many cases. Then you have, but the less epidemic and so on, like 800 persons. Okay, so yeah, so you see that the COVID is much more important. Okay, much more important. I mean, there were times where 800 people died in a single day. Okay? So. Okay, I don't know even why we are discussing this. Okay, it was because of the functional forms for malaria, so I just went thinking. Yeah.
So, I think it isn't really clear to me what we do with the model after feeding to data. Like, what are the quantitative methods to validate the model?
Okay, models are rarely validated because of the unknown parameters. Okay, so okay, let me go back to COVID and talk of something that I did. Okay, so with the group, so there was this outbreak of new variant in Amazonas, Manaus. Okay, super explosion of cases and so on. Oh, this is the gamma variant. Okay, so we wrote a model which is has to include a lot of things. Has to have hospitalized people, people that die from it, in not only SIR. Okay, you need because these are usually not observable. I is not observable. The number of people that are infected at a given time, you don't know, because there are symptomatic cases. You don't have how to measure this. What you can measure is usually hospitalizations. So the hospitalization rate, number of new hospitalized per person per day or per week, it's what is really observable. So you need a compartment for hospitalized people. Okay? Then you have the dead people, and then you have to put classes of age, because all these parameters can depend if the person is old or not, or if it's young. You can do all of this. Okay? And many of the parameters are known from, there were so many studies and so on. So we wrote that, and we actually, what we did is, we have left some parameters free, which is the transmission rate, okay, the infectious rate, and we also left free the number of people that were that had previous infection, that had before this wave, was also, and the start that date that the variant is introduced in the population. Okay, the starting date of the variant. So we left these things and we fitted the model to the data. So therefore, what we got how transmissible is the variant, so that we could therefore say that we had a variant that was, in this case, 2.5 times more infectious than the previous variant. Okay, so that's a use of the models. Okay, so it's when you see people, this variant is more infectious and this one is blah 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 and so on. What they are doing is usually a model that can be large, small. There are many models, and then you infer, for instance, the infection rate. Okay, and to know if it. So, but we also had in this model, this is interesting because you see that there had been also so many cases before. So the susceptible population should be small. How come is this happening to have such a burst of cases when the people were supposed to be immune? Okay, so we could also calculate the percentage, but okay, you could even say maybe it's just immunity loss, not higher transmissibility. Okay, okay, so we had both. We fitted the transmissibility and also the number of reinfections. Okay, and then you do a modeling which is not only, I mean, you do some data fitting which is not so trivial and which will run on a very good computer for a whole day, because you are searching the space of parameters, okay, and finding the most likely values. And it turned out that we got the transmissibility was 2.5 times the transmissibility of the resident one, and that 28, more or less 28% of the infections were reinfections. And that at the starting point of the new introduction of the variable, the persons that had previously been infected and had some immunity were 78% of the population of Manaus. Okay, it turned out that there was a group of people that, not collaborators, but people that we talk all the time, a group from Esther Sabino, which actually has, using blood donors in Manaus, has calculated the number of cases which were reinfections, and you obtain it 30%. Okay? Because you have people that regularly go there, you know this person has already antibodies to the previous variant, the original one, and then the same person you get again sick, and then he has been reinfected or not. Okay, so that it's a proxy. And it's obviously not a controlled measurement because it has bias in the sense that the people that are donors. Okay, maybe all the people that are donors are also people that, I don't know, use mask or whatever. Okay, so there are uncontrolled bias. So it's always open to discussion. But anyhow, but very nice because what they found is actually that our result was pretty much close to what they obtained. Okay, between the error bars was okay. So that's the thing that you can do with the models in this case. Okay, so and many things are done in this way in epidemiology. Okay. The parameter, the value of the parameter itself, it's very important, okay? And so knowing the parameter. So then you could also predict, but so predictions turned out to be very difficult with the COVID-19 epidemic, very difficult. It failed all the time. Nobody would have thought that variants would appear in such a short time and that there would be many, many of them. No people working with virologists working with coronaviruses would have predicted this space of emergence of new variables. So that, you could do models and predict and so on, but yeah, you didn't take into account variance. And loss of immunity. Loss of immunity is very difficult. There should be some typical time for immunity to fade, okay? So, but how do you know it? Okay, you have to I mean, you cannot predict, you cannot write a model and predict many things if you don't have enough time to have at least some idea of the time it takes for a person to become susceptible again, or partially susceptible, and so on. So this is also something that it's difficult. So what turned out with the COVID things is that kind of complex diseases like this are modeling has a different status. It's not so predictive. But that is okay with modeling. Modeling is you do some scenarios, okay? So you have a situation that you can model, and then you have scenarios for the future, and your model can say if there are more dangerous cases, or if for instance, reopening schools, okay? That could be, you can have many scenarios and make predictions on the short time scale for the scenarios. So for instance, reopening completely the schools, or only 50% of the students come, or only 10%, or on 80%. What happens if we try to include masks or interventions and so on? This kind of thing helps you to say to look at what would be, for instance, a safe range to reopen schools, okay? Because there are nonlinear things, and for instance, with data from also from Manaus, we made an exercise because the school had already reopened. Okay, but you could also make an exercise and say how if I open only 10% of the students come back, well, it's really safe. But it's not just proportional; it's non-linear, and over 40, then you get a very high number of cases, 40%. Okay, so there was a range of values where it would be okay to reopen under certain conditions, like masks and so on. And well, obviously no decision maker takes this into account. Here in Brazil, it's impossible. I mean, you have to speak with the governor of Amazonas. He will not work. Not even with this health secretary of Manaus, or even São Paulo. We had a fight with people from São Paulo sometimes. So yeah, so the model is this kind of thing. It's no magic. That the model will do no magic, okay? But it's also better than not having anything, right? Not having any model puts you in ecology and in epidemiology. And even if model could be also only conceptual, not so mathematical, and so on, or having models, not having models, would give you only a kind of lots of data that you don't know, you don't get actual knowledge of the data. You have the data. I mean, you could go for machine learning things, it's like, okay, which is a different story. But you have the data, and you have to interpret the data. What is the data telling you? You have to have some concepts that are clear. Now, even if you do a lot of things which is not mathematical, but having data means you have quantities that have been measured, and so on. Somehow you have to have maybe in a statistical model. You could say, no, I don't have this kind of dynamics. I have just correlation between variables, and determine if the correlations have something to do with causality. So in this case, you don't have to dynamics like this. You have a statistical model, and you have a way of inferring causality, which is difficult, but sometimes you can do this. So this is also a statistical model that will allow you to have access to information that's important, which maybe will allow you to then write models of dynamics, because all of this includes time, okay? So it's a model of dynamics here. Okay? Sometimes you don't have this. Sometimes you have only only, you don't have the time evolution, but you have correlations, spatial or maybe time also. Yeah. So, okay, anything else? Any other questions? Okay, so that's it for today, and Monday we will go back to consumer resources and see what we can do better with consumer resources than with competition things, okay? And then we will have the opportunity to discuss also, at a certain point, the concept of niche, which is a complicated concept in ecology, but it's nice to discuss in view of the consumer resource dynamics. Okay, so thank you.